first and foremost, insulin is huge, right now. We talked a little bit about last time about a protein called glycogen-like protein, right? Which GLP-1, which is used a lot nowadays as an injection to help people lose weight. Yeah.
Is it like Mounjaro?
Yeah, I think it is Mounjaro. It's at least one of the names of one of them, right? And so they're you can get them ordered online now, and it really does help with weight loss, but it also helps with insulin regulation. So, my wife is actually she's in a pharmacy that specializes with mental illness, and they're using it and to treat mental illness also. So it does change brain chemistry a lot. So it's one thing you want to think about long term. But it does work really well. So, and not only does it cause the brain to think you're full, or it stimulates what's called the satiety center in the brain that says, oh, I'm full, my stomach's full. That helps you control calorie intake. I guess it helps with sweet cravings and that kind of stuff. So this is basically how insulin and glucagon work as antagonists. And this is why I picked this, not because it's really complicated, which it is, but because this is how a lot of antagonists work when you have two hormones that do opposite things, right? They are going to ultimately activate opposite enzymes, and it's going to be phosphorylation. So this is just to hammer into your heads. Hopefully the hammering is real, right? Is that kinases turn things on and off. And so does another enzyme called a phosphatase, which is the opposite of what a kinase does, right? So kinases phosphorylate things, phosphatases remove phosphates. So it makes sense that one of the hormones, insulin, is going to activate phosphatases, and the antagonist is going to activate a kinase, right? Also, we have two enzymes. The liver, if we're going to talk about the liver, and then I'll mention we can add body cells to this whole picture. The liver is our major glycogen storage site for glycogen that's usable by our bodies. So you can store glycogen there, and then when you're fasting, you can release or break glycogen down to release glucose. And so it's kind of a carbohydrate storage area. Everybody knows glycogen is just a polymer of glucose, right? You just hook a bunch of glucose molecules together. And hepatocytes are responsible for storing glucose that we can use later when we are in a fasting state. So the liver will respond to both of these hormones, insulin and glucagon. Notice I also put epinephrine down here because it does the same exact thing as you saw. Did you guys go back and look at that mechanism? You may or may not have, right? But remember, there's that one mechanism that I showed you how epinephrine or glucagon causes glucose release from the liver. And ultimately, if you look at that mechanism in that second or third lecture, this is what you're going to activate, right? When you release epinephrine and you get that burst of glucose, that flight or fight response, or when glucagon's released in a fasting situation, you're going to activate a receptor that is an adenylate cyclase G-coupled receptor that activates a bunch of kinases, right? And it's actually protein kinase A, which will then activate this kinase and this kinase and this kinase, which ultimately will turn on phosphorylase kinase, which phosphorylase is the enzyme that breaks down glycogen, okay? So when I turn that on with kinases, notice phosphorylase kinase is going to phosphorylate glycogen phosphorylase, which turns it on, right? And you start breaking down glycogen into glucose. Now, if glycogen turns on glycogen phosphorylase by phosphorylating it, then glycogen or epinephrine, when it phosphorylates the other enzyme, it's going to turn it off, right? So notice over here, the same mechanism, right? Glycogen binding will also activate cyclic AMP, protein kinase A. Notice it phosphorylates glycogen synthase, which turns it off. That way, you're not building glycogen when you're trying to break it down, right? You're actually turned off glycogen synthesis, and you're liberating glycogen breakdown, right? Getting glycogen breakdown. So the first thing, when I go to memorize this, what I would do is be like, all right, so when I phosphorylate glycogen synthase, what does it do? It turns it off, right? When I phosphorylate glycogen phosphorylase, it turns it off. So first, know what phosphorylation does, because that's what I've taught you about kinases, right? That they're on and off switches. So if phosphorylating glycogen synthase turns it off, when I want to turn it on, what do I need to do? Remove the phosphate group, right? And that's what insulin does. Notice insulin only activates phosphatases. And in particular, the phosphatase here is called PP1. This is the worst thing about molecular mechanisms, is there's 5,000 enzymes, right? And then they abbreviate all of them. And you got to read a paper and write them all down, right? And so I'll give you the names of all these as we go through it. But all the names are listed underneath in the notes of the slide, right? I'll probably write them down and throw them on the PowerPoint when I do it also. But PP1 is the specific phosphatase. You can imagine there are lots of phosphatases out there that actually will remove phosphate groups. So, and if you look over here at this really complicated mechanism, notice PP1's here and PP1's there, right? Don't worry about G. It's just a variation of what the protein looks like. We'll just keep it simple and call it PP1. So these are phosphatases that are removing phosphate groups, right? So when I want to turn on glycogen production, I'm going to turn on glycogen synthase, right? And I'm going to do that with insulin, right? And insulin, when it's released, your body's saying, hey, a bunch of sugar is about to enter the blood, take up anything excess and store it, right? Your body cells will also respond to insulin. But in the liver, what that's going to do is when insulin binds to its insulin receptor, it's going to turn on a cascade of kinases that eventually lead to the activation of PP1, which will activate protein phosphatase 1 and remove the phosphate group from this and turn it on, right? And now glycogen synthase starts making glycogen. So if you look at this mechanism, let's find our way to PP1, right? That's what we want to do. Let's do that first. Right? And it's a specialized tyrosine kinase receptor. It's actually called a dimer, right? So a dimer is something that has two mirror image protein subunits. And they're actually separated in the membrane. And when insulin comes in, it brings the receptors together, and that brings the two subunits over here together. And when insulin brings these two, I almost think of them as ladders, right? Brings the two parts of the ladder together, then phosphate rungs, disulfide bonds are made between them, holding the receptor together. And that is going to bring the tails here, which actually span the membrane, in contact with one another. And so binding insulin brings the dimer together so that the tyrosine kinase, the two subunits of the tyrosine kinase touch, and now they work. And so what does tyrosine kinase do? It phosphorylates tyrosine groups. Well, guess what? You know what it phosphorylates? It phosphorylates the actual receptor, because this receptor has a bunch of tyrosine groups on it. And so it's phosphorylating itself many times. And so when you have an active dimer that's together with insulin attached, you're going to add a bunch of phosphate groups to the tail, which lights it up, right? And then a number of proteins can bind to it. So this is what's cool about a tyrosine kinase. If I light it all up with phosphate groups, then I can turn on multiple proteins at once at the receptor, all right? And so these other proteins are just floating around. And they become activated because, like we know, phosphates turn things on. And so we can follow a couple different proteins here. Since we're talking about turning on glycogen synthase, we ultimately want to remove phosphate groups from it. So we want to turn on PP1, right? And so let's do that. One of the proteins here that's going to interact is a middleman that's called IRS1. All right, IRS1, which is insulin. And then the R stands for receptor. And then the S is substrate. So insulin receptor substrate, obviously number one, because there are others. Oh, damn, I just drew on my shirt. Insulin receptor substrate 1, or IRS1. Right? Notice it has combined to the phosphate groups that are phosphorylating the tyrosine groups. That will allow another protein to dock that's called PI3K, right? PI3K, which is phosphoinositol 1 kinase. Is that 1? There are 3, sorry. There are many of them. 3 kinase, right? So phosphoinositol. Remember, inositol is a fatty acid, right? And where are those found? They're found in phospholipids, right? They're found in phospholipids. Remember, IP3 is phosphoinositol, right? Triphosphate. We're going to kind of do a similar thing here. So notice what we're doing is we're bringing this middleman docking protein on board so we can turn on this kinase, right? And what it does is it phosphorylates phosphoinositol, which is right here. As you can see, it's sitting down here in the membrane. We add a phosphate group, and we go basically from with something called PIP2. And what this enzyme does is add a phosphate group to it, so it becomes PIP3, right? Basically, we have inositol biphosphate. We add a phosphate group to it to make it triphosphate. Remember, IP3 is a triphosphate. For some reason, when you add 3 phosphates to a fatty acid, it really energizes it. And so if we add a third phosphate to this, we get PIP3. And PIP3 can then bind to an inactive kinase called protein kinase B. It also has like 5 other names. So if you look up this mechanism, it's got a bunch of different names. But we'll call it protein kinase B. So activating IRS1 activates PI3K, which then turns PIP2 into PIP3, and PIP3 turns on protein kinase B. All right? So we know what protein kinase B is going to do. It's going to phosphorylate something, right? So, and what it phosphorylates is this protein right here, glycogen synthase kinase. So glycogen synthase kinase is a kinase that's going to phosphorylate glycogen synthase. Remember, phosphorylating glycogen synthase turns it off, right? So notice here, if I'm glucagon or epinephrine, I can activate protein kinase A and turn glycogen synthase off. But notice in the liver cell, glycogen synthase is default off because of glycogen synthase kinase. If it's on, it is phosphorylating glycogen synthase and preventing it, right, from working. It's keeping it off. So if insulin binds, the first thing it does is turn off glycogen synthase kinase. And now you can't phosphorylate this anymore, right? It's also going to activate, if you do that, you turn that off. Now you can't phosphorylate this anymore. Then you remove the phosphates, right, by activating PIP1, okay? Notice here, PIP1, what do we do here? We remove the phosphate group. Here's PIP1, removing the phosphate group and turning glycogen synthase on. So insulin turns off the kinase and turns on glycogen synthase by removing a phosphate group, right? How do we turn this on? I don't show you that, right? I just show you this positive. I'm going to turn on the kinase on it, right? Here's the kinase right here. So this can bind directly to the receptor, or the receptor, once it's phosphorylated, turns on this big protein that's called insulin substrate protein kinase, right? Insulin substrate protein kinase. which then will phosphorylate PIP1, turning it on and allowing it to activate glycogen synthase. Ultimately, both of these, turning off the kinase that keeps glycogen synthase off and removing the phosphate groups with this kinase, ISPK, will turn on glycogen synthase and you'll start making glycogen, right? Believe it or not, PIP1 also inhibits protein kinase A, which makes sense. And I didn't draw a line there, but it will also inhibit that. Insulin will also do this in all body cells out there, including the liver. If I want to get glucose into a cell, I need a channel, right? And so what insulin does is it tells body cells, hey, glucose is available, so insert channels. So this is why if you have type 1 diabetes and you're not making insulin, your cells have no idea that there's glucose available. And you could have the highest blood sugar in the whole world, and your cells have no idea because there's no insulin saying, hey, there's glucose. So obviously, normal body cells can't sense glucose by themselves. They need help, right? And that's why the pancreas is so important, because you need insulin. Or if you downregulate the receptor, you become insulin intolerant, right? Or what they call it insulin tolerance. So some people are genetically that way. They just downregulate the receptor, which makes them more susceptible to type 2 diabetes, which is a receptor thing. You're not producing enough receptor. Does that make sense? Which could increase your blood sugar overall. But once insulin binds to its receptors in body cells, notice it also can activate this protein here called CBL, which is, that name doesn't make as much sense, but it's dicyanocobanamide. It's a freaking huge word, I know. What this is it's a vitamin B derivative. This is why one of the reasons vitamin B is really important. So, the coban part of it is the vitamin B. It's actually a pigment, believe it or not. Cyano is blue, right? So this protein here is a kinase also, right? So it has the ability to activate kinases that ultimately lead to the phosphorylation. So notice you phosphorylate dicyanocobanamide, and that will turn on phosphates that eventually lead to you phosphorylating vesicles with glue transporters in them. And, if I light up the vesicle, then kinesin or dynein can grab onto it and walk it, right? Walk it to the surface. And then I will insert those channels into the membrane, and I can start taking glucose out. So glucose channels are interesting. They're added and taken out of the membrane liberally. That is, once they're added, they're only there temporarily, and then they're dragged out again, brought back into a vesicle or recycled, and they're used in a membrane recycling routine that means the glucose channels don't stay very long in the membrane. They're actually removed really quickly in most body cells. So I got to continue to add them if I want to remove glucose from the interstitial fluid, and that's what insulin does. It's telling you in this pathway, whether you're a liver cell or a body cell, that you need to insert glucose transporters. Obviously, the liver needs glucose in order to build the glycogen. So this happens in liver cells, and so does this process. Insulin does a whole bunch of other things in cells. You should just look up a picture just for fun of what insulin does in cells and find an image, and you'll be like, whoa, there's all these things it does. It helps build tissues. It turns on protein synthesis. It increases lipid production. It does all kinds of weird stuff. So, and so there are a bunch of other proteins in different body cells that can bind to the insulin receptor because of the phosphorylated tyrosine groups. Now notice, I kind of mentioned that ISPK, this insulin substrate protein kinase, can phosphorylate protein phosphatase 1 and allow it to remove the phosphate group so that glycogen synthase is activated, right? So that would be this part here. But notice what also what it does. protein kinase 1. It also is going to remove the phosphate group from phosphorylase kinase. So if I remove the phosphate group from phosphorylase kinase, which is going to phosphorylate glycogen phosphorylase, that would turn it on, right? Over here, see that? So if I inhibit that kinase, then I'm not going to turn this on, right? Also, my protein phosphatase 1 will remove the phosphate group from here. So notice this word down here, phosphorylase kinase. Phosphorylase kinase, right? Glucagon and epinephrine use cyclic AMP to turn this on, which will then phosphorylate glycogen phosphorylase, which starts breaking glycogen down. So if I inhibit that with protein phosphatase 1, then I can't turn it on anymore, right? So notice protein phosphatase 1 not only removes phosphate groups from glycogen phosphorylase, turning it off directly, but it also inhibits, that's what that line stands for there, phosphorylase kinase, which again turns that off so that I can build glycogen. Whew. What the hell? That's you gotta work your way through it, right? This is why I don't teach you thousands of them, because now you see why endocrinologists are really brilliant, right? They got to be really smart or really geeky and like to learn this stuff, right? I mean, anybody can learn it. You just you want to dive into it. So, but this is what happens when you have antagonists. It's a little simpler when you got one hormone just doing something, right? But you're working on one organ with two different hormones that are doing opposite things. You got a bunch of different proteins involved. Make sense? All right. So let's look at a couple of other hormones. I want to do, and that's really the big mechanism that I'll teach you. The other mechanism I'll teach you in this particular lecture is thyroid hormone production and its stimulation, because thyroid hormone, again, really important in basal metabolic rate in our keeping our body temperature warm and basic metabolism, and hypo and hyperthyroidism are really common, right? And the medical profession. And thyroid hormone is a huge synergist, and that's why I also like to talk about it, because not only are hormones important, but they actually affect each other. And so, for example, if you have thyroid problems, you have a really hard time getting pregnant. If you're a female, all four of you are females, right? The reason for that is because thyroid hormone works with sex hormones to prep the reproductive tract. And so without it, the sex hormones are really poor at doing their job. So it's what I mean by a synergist. It actually helps. And it has very little to do directly with the reproductive tract, but it helps those hormones do their job. And so we'll talk about that as we get into talking about thyroid hormone. So long distance communication. So there are two ways to do it, right? Two major ways we've already talked about: neurons, and then what we call classical endocrine organs. So there are lots of them, and notice they're all spread out. So this picture here in the human, or you can look at any real vertebrate, any mammal, and other terrestrial vertebrate, has most of these structures that are responsible for releasing hormones. Now, what I want you to notice up here is here's the central nervous system, right? It's the brain and part of the upper spinal cord. And you're like, those are where your neurons are. But remember, neurons can also release hormones, right? Remember the definition: neurotransmitter, nerve to one cell, specific cell, or a group of cells. A hormone you dump into the blood, right? And so the nervous system can produce both neurotransmitters and neurohormones. Ultimately, the nervous system can control most endocrine glands. So in reality, homeostasis is really a central nervous system kind of monitoring or control center. But the central nervous system does control a gland called the pituitary gland, which is often called the master gland in endocrinology. because it controls so many other endocrine glands. And so I always like to teach, what the pituitary gland does and kind of how the brain controls it. Because it often will release hormones that will control other glands. And those hormones are called tropins. All right. And so most of the hormones of the anterior pituitary are going to be called tropins. Right? So, and I mentioned that word last time, I think. So tropins are hormones that control other endocrine glands. Okay. So thyroid stimulating hormone is also called thyrotropin. All right, that's how I learned it. Old school word, thyrotropin, because it controls the thyroid gland. So we'll talk about those. The one thing that I didn't mention, and we've talked a little bit about immunity, is that the immune system releases these chemicals, chemical communicators that can often be called local paracrine hormones, or in some instances, there's a lot of argument, there's a lot of gray area, but a lot of argument that some of these inflammatory molecules can be endocrine in function, that is, affect lots of organs all at once. And they're called cytokines. So again, if you look up a picture type in Google, what do cytokines control? And go to images and look at images and pull up a map of what all the cytokines do, and it'll absolutely blow you away. Immunologists are incredibly brilliant people also, because they have to keep track of so many things. There are tons of different cytokines released by tons of different cells that control immunity and inflammation. Most of them, in my opinion, my humble opinion, are paracrine type of molecules. They control things locally, local inflammation, local immune response. But many of them can be released into the blood and start affecting organs dramatically. That's all I'm going to say about it, that they are cytokines. So those are kind of the communication molecules of the immune system. Okay, we did this picture, but I always remember to throw it in there, the difference between an endocrine and an exocrine gland, right? So to give you an image. So exocrine glands and endocrine glands are both epithelial in origin mostly, right? That is, they come from an epithelium. And most glands in your body are epithelial, right? And these epithelial cells produce a product and they dump it somewhere. In an exocrine gland, they dump it into a duct. In the endocrine glands, they dump it into the interstitial space and it enters a blood vessel. Because of this, most of your endocrine glands are heavily vascularized. Right? That term vascularization, I've used it, right? Heavily vascularized. That means lots of blood vessels. They have lots of capillaries. A lot more capillaries than a little ball of cells should have. Because what do? They dump stuff into the blood, right? And so that's what vascularization means. There's a lot of blood vessels that grow into the organ so that the endocrine gland has somewhere to dump, right, its product into. Okay, so here's the pituitary. So if you zoom in on the pituitary gland grows off of a part of the brain, sits in a little cavity of the skull, in the ethmoid bone called sella turcica, if you ever taken anatomy, right? But it is literally, at least the posterior lobe is an outgrowth of the brain. It develops as a bunch of nerves that grow and migrate and grow right out of the lower brain, out of the region of the brain that's called the hypothalamus, right? So the hypothalamus sits on the bottom of kind of on the ventral surface of the brain, just underneath the thalamus, which is the bridge between the cerebrum and other parts of the brain. The hypothalamus is hollow. It's kind of hollow. It's got what's called the third ventricle. It's got a space that's filled with cerebrospinal fluid. And the pituitary, the posterior pituitary at least, grows right out of it. The anterior pituitary has no connection to the hypothalamus. When I say that, no direct connection. Okay? That is, it's not the anterior pituitary or lobe there are two lobes. They call them the posterior pituitary lobe and the anterior pituitary lobe, or a lot of people call it posterior and anterior pituitary glands. The anterior part of this is actually epithelial tissue. And it grew out of the roof of the mouth during development, right? So if you ever take a developmental course and they talk about development or cephalization in the head, there is a period of time when a little group of cells bud off of the roof of the mouth, and it's called Rathke's pouch is the name they give to it. You don't even know it. That this little pouch forms, and then the cells bud off, and they divide, and they migrate, and they attach themselves to the posterior pituitary. And so they are actually epithelial cells, group epithelial cells, that actually secrete most of the hormones of the pituitary gland. So I call the pituitary gland the master gland. It's really the anterior pituitary that is the master part here. It has a bunch of different cells that produce a bunch of different hormones. The posterior pituitary does produce hormones, but only two of them. At least, well, the two we understand. How's that? The best. And because it's a direct outgrowth of the hypothalamus, it's actually nervous tissue. So it's actually made up of a bunch of axons of nerves that end in the posterior pituitary and dump their product, their neurohormone, into the blood. So in this instance, think about it. The hypothalamus is directly in control of the secretion of the posterior pituitary because it basically sends its axons down into it. And then just when they're stimulated, it releases the neurotransmitter, right? But the hypothalamus is also in control of the anterior pituitary with no direct connection. So then the question becomes how, right? How is that possible? And the hypothalamus produces hormones that it dumps into a capillary bed right here at the base of the stalk of the pituitary gland. Think of this as a grape or a cherry. It has a long stem. They call it the pituitary stalk. And then it has the swollen ball, the fruits, right? That's kind of hanging off of the brain. This first capillary bed is right against the base of the hypothalamus. And there are nerves in the hypothalamus that their axons end right next to this capillary bed, and they dump their product into that capillary bed, which then travels through two blood vessels that are called portal vessels. Okay? These portal vessels connect this capillary bed directly to another capillary bed, which is sitting over the anterior pituitary. So a portal vessel is weird. A portal vessel, remember, most capillary beds, arterial feeds it and a venial drains it. Right, a group of venials. Here we have arterioles delivering blood, but no venials. We have two portal vessels that connect it to another capillary bed. And then venials, right, after that. So when two capillary beds are connected in tandem, they're called portal vessels. So this is the hypothalamic anterior pituitary portal vessels. The best known portal vessels in the body are the hepatic portal vessels that connect the capillaries of the digestive tract directly to the liver before you pass blood into circulation. So everything from the digestive tract has to go through the liver first before it enters circulation. This is the other place. This way, the hypothalamus can send a hormone right to the anterior pituitary, and it doesn't have to circulate all the way through the body before it comes back, right? And that's how you get indirect control through two capillary beds and a portal vessel, for a group of portal vessels. Does that make sense? Now, the hypothalamus, the hormones are called hypothalamic releasing and inhibiting hormones. Right? So if they're a releasing hormone, they're going to stimulate the cells to release their product. If they're an inhibiting hormone, they're going to inhibit those cells from releasing their product. Right? And so inhibiting and releasing hormones, and you'll hear me mention those as we go through this. Now, here are the major hormones from the anterior pituitary. And these are the ones you're going to learn. So we got prolactin, which is the only hormone that's not a trophic. All right, prolactin is the only hormone from the anterior pituitary that is not a tropin. That is, it doesn't activate another endocrine gland, but it does activate a gland, but it's an exocrine gland, right? And so prolactin in mammals activates the mammary glands, right? And gets them ready to produce milk, right? So it's going to cause differentiation and division of the lactiferous ducts and the lactiferous glands of the mammary glands so that they are enlarged, typically towards the end of the monthly cycle in a mammal, so that the breast tissue is ready to go if pregnancy were to happen. We also know prolactin is important in other animals, right? Other vertebrates that aren't mammals. Remember we talked about a fish that actually changed the gills, saltwater, remember, to freshwater? So that's the only one. Pro means to activate. Lactin means milk production, right? So that's why the hormone probably should have been called something different in fish, but it was first discovered in mammals. And so the name sticks, even though fish don't produce milk. That's not true. Sharks produce milk, but it's a different type of milk. Bony fish don't produce milk. So that's the only non-tropin. The rest of these are tropins, right? And so they're going to affect other endocrine glands. Now, I know growth hormone here says that it activates the musculoskeletal system. Growth hormone is responsible for growth and maintenance of the body tissues, but it does stimulate the liver to release hormones. All right. So, growth hormone is also a tropin, even though it's often not taught as a tropin, because it causes the liver to release insulin-like growth factor. All right. Insulin-like growth factor, which is going to kind of synergize with insulin and allow for growth in babies, pre-adolescents, right? But it also helps with our tissue maintenance, right? Our normal tissue maintenance. So GH is growth hormone. TSH is thyroid stimulating hormone. And thyroid stimulating hormone is going to control the thyroid gland directly and cause it to release thyroid hormone. ACTH is adrenal corticotropin hormone, or it's going to control the adrenal cortex, which is, remember, the adrenal glands. We know they release aldosterone already, right? But there's a bunch of other hormones that the adrenal glands release. ACTH usually is involved in the release of cortisol, which is the stress hormone, which I relieved Faith of today. Right? At least till next week. I'm sure you have plenty of it for your other tests. The last one are called gonadotropins. So the gonadotropins include two hormones called LH and FSH. So gonado, they affect the gonads. Gonads, ovaries, and testes produce the sex hormones, right? Estrogen and testosterone. And so FH and LSH stimulates not only the gamete production and maintenance of the secondary sex characteristics, but they also cause those hormones to, those organs, the ovaries and testes, to release their hormones, which are estrogen and testosterone. So I give you a couple pictures here. I like to lecture on this slide and then show you images. Right? So this is how the hypothalamus directly controls the posterior pituitary. It's real simple. Here's your cell body and your dendrites. They're way up here in the hypothalamus, the wall of the hypothalamus. They send their axons all the way down through the stalk, and the axon ends in the posterior pituitary. And then it just dumps the hormone directly into the blood supply. So they're neurohormones. This is an image. If I were to blow up an image here, these are axon terminals. This is the wall of the blood vessel. So they just end and they just dump their hormone directly into the blood. And so there are two hormones from the posterior pituitary One of them we already talked about. Remember ADH, antidiuretic hormone. And then the other one is called vasotocin. ADH is also called vasopressin. It's the name that you might see somewhere. That's how I learned it. Because it has an effect on blood pressure, right? Remember, when ADH is present, it causes the collecting deficit of more water, increasing blood volume, and raising blood pressure, right? Vasotocin causes smooth muscle contraction. So, in females, that's usually how it's taught. In females, it is what causes contraction during birth. Yeah, you love that, right? That's you the only ones who face women because you've given birth. Yeah, yes, it's females are incredible. Yeah, with the stress that the baby puts them through. Vasotocin is also released during ejaculation, both males and females, believe it or not. But it's mostly, right, that's the fun part. The horrible part is, but it's not horrible. It's not pleasant. How the hell would I do it, right? I watched my wife give birth to 4 babies, and I'm immensely respectful. So. She wouldn't let me video it. It's funny. So when our doctor was awesome, my wife's gynecologist in Ohio was awesome. She was a very nerdy person. When she found out I was a biologist that studied reproduction, when Allie was born and my wife delivered her placenta, she threw it on the table and opened it up, and then she was like, "Come over here, Kevin." And she started we were talking about the placenta, and my wife's bleeding everywhere, "Hey, I just gave birth. You want to take care of me?" The doctor's, "Oh, I'm sorry." It was really fun. Got in trouble for that. I was like, "I didn't have anything to do with it. You wanted to show, man." Blood doesn't gross me out, so I could have been a surgeon. All right, so this is one of my favorite pictures, histological pictures. So this gives you a really good idea of what this is a mouse brain, so that's why you can see it. They just sectioned through the head, frontal section like this, until they got to the hypothalamus. And what they did is they removed all the calcium from the bones. You can use kind of a leaching agent to remove the calcium salts. And so that just leaves kind of the collagen and bone cells left. And this is the little depression that the pituitary actually sits in. This is all bone tissue out here. So this is all what they call the ethmoid bone, part of the ethmoid bone, body of the ethmoid bone. It's one of the bones in the skull. And the pituitary sits right in there really nicely. It's pretty cool, actually. The other thing that's really nice about this shot is you can see the stalk really good. And notice the posterior pituitary, you can always tell which part is the posterior pituitary because it has the same staining properties as the brain tissue above it. See that? So it's the same color, light pink, same kind of texture. Whereas the anterior pituitary is very different in its color and texture as far as what it looks like. So you can see it's darker staining in this instance. The hypothalamus is right here. This is the hypothalamus. It's not real, awe-inspiring when you look at it. It's a very thin, section of tissue, but it's like the major controller of everything that's autocrine, or automatic or part of the autonomic nervous system in the body. This big white space is purpose. It is a purposeful space. It's not this person or this mouse had no brain, like me, no cells, right? Small brain. This is the third ventricle. It's the actual space that holds the cerebrospinal fluid. So I don't know if you know this. I don't know how much anatomy you guys have. So the brain is it's hollow. It's got spaces in it that are filled with cerebrospinal fluid. The third ventricle receives cerebrospinal fluid from the 2 lateral ventricles, right, which are up in the cerebrum. Little anatomy lesson, right? Now, if you look at the posterior pituitary, it'll be all axons. If you look at the anterior pituitary, it'll be a bunch of cells and get a lot of capillaries. So this is a human, or it might be primate actually. It's a primate pituitary gland that shows you the pituitary stalk, and this is the anterior pituitary. And these are the cells, right? So there's no striations or axons in there. And you can see all the little white stripes, right? All the little white spaces, those are capillaries. So they not only deliver the hormone from the hypothalamus, but they also take the hormones away, right, from the cells. Now, I know us biologists are horrible. So we use tropins as our name for a hormone that controls another hormone, right? The cells of the anterior pituitary are called trophs, right? Which always confused me. They're called trophs. Trophs release a lot of tropins. So trophs means cell, right? Cell, it really means unit. So trophs are cells. And there are obviously a few different types of trophs that are found in the anterior pituitary. All of those are epithelial in origin. And so here are the troph cells. Now, they all look alike if you stain them with normal stains. Right? They all look alike if you stain them with normal stains. So the way you can get them to light up is to actually do a technique where you label the hormone, right? You label the hormone. Most of these hormones are proteins, so it's really easy to make an antibody for them. And if you make an antibody for them, you can expose the mouse in the blood with the antibody, and the antibody will travel to the brain, and antibodies can enter cells, and it'll stick to the cells that produce the antibody. Because remember, antibodies are very specific to the protein they bind to, right? The antigen. And so this type of microscopy, and I don't know if you've ever heard of this before, it's called immunocytochemistry. It's when you use the immune system, or the properties of the immune system, to light up something in a cell. And so we use what's called a primary antibody. And so, for example, the thyrotrophs. Thyrotrophs produce the protein thyroid stimulating hormone, or what's called thyrotropin. Okay? And it's really easy to get them to light up. What you do is you produce an antibody for TSH, right? You inject it into the mouse. It'll travel through the circulation into the pituitary, and it will then go into the thyrotrophs, bind to thyrotropin. What do you want? They bind to thyrotropin, right, or TSH. Then you use a secondary antibody to bind to the primary, and it will light up. Okay. In this instance, that secondary antibody can have a fluorescent tag on it and light up yellow or green or blue. This is a cheaper way to do it. You use an secondary antibody that has an enzyme on it that will take a compound and make a pigment. And here it's brown. And the pigment's called horseradish peroxidase. It actually takes horseradish and then turns it into a brown substance. It oxidizes it. That's what peroxidase does. And it turns the cells brown. So only these cells that are brown here, not yellow, are thyrotrophs. Over here, the ones that are brown are gonadotrophs. Over here, the ones that are brown are corticotrophs, and so on. The majority of the cells of the anterior pituitary are somatotrophs. All right, somatotrophs. They release growth hormone. Growth hormone is basically controlled by concentration. All right, and so as a mammal, you simply release bursts of it all day long and keep it at a pretty normal level within the blood so that it can do its job in what's called animalism, in building things in your body. Obviously, when you're young, you release a ton of it. So giantism, you've seen people that are humongous, right? They're really tall. They obviously produce, particularly when they're young, if they're really big, when they're 8 years old and they're not, they've either entered puberty early, or they might, some of the tallest people in the world had a tumor of the somatotrophs, and they had too many somatotrophs, and they were too tall, too big. So you got genetic control of how much of that you release. Most growth in big boys, at least, and girls, is controlled by estrogen and testosterone. Right? which caused massive load. But these are all showing, the cells lit up in different areas of the pituitary, anterior pituitary. So this figure here is because I've kind of already talked about it. This figure here gives you your 5 different trope cells and what they produce, right? All of these are tropins except for prolactin, right? So all of these tropes produce tropins except for lactotropes. Someday somebody will probably provide us some data that suggests prolactin does something hormonal, I would assume eventually, right? But as of now, it just initiates milk production in the female. So somatotropes produce growth hormone. Growth hormone stimulates general body growth and regulates metabolism. It also causes the liver to release insulin-like growth factor. They're called ISGs. Insulin-like insulin-like growth factors, ILGFs, right? Or sometimes they're just called IGFs. Gosh, I can't write. IGFs. And there'll be like 1, 2, 3, there's a bunch of different types. Insulin-like growth factors are what are released when people produce really big babies. They produce too many of these insulin-like growth factors, which can also cause insulin release and then cause pregnancy-based diabetes. Sometimes it's really bad. You get the collapse of the pancreas after pregnancy, and then they're full-blown diabetics. But that's what IGFs do, is they help with growth. They also mimic insulin. So these IGFs are produced by the liver. And so the somatotropes, the tropin part of it is the liver is its target. Thyrotropes produce thyroid stimulating hormone, or what's called thyrotropin, and it stimulates the thyroid gland to secrete and produce thyroid hormones. Gonadotropes produce gonadotropins, FSH and LH. Follicle stimulating hormone was first named because the hormone was found to cause egg development in the ovaries. Follicle stimulating hormone is also really important in males. It is what initiates sperm production. So it's also really important in males. Luteinizing hormone was first discovered to cause the secretion of testosterone, but it also stimulates estrogens in the ovaries. Believe it or not, females produce testosterone first and then convert it to estradiol. So one of the animals in the world that produces the most testosterone is the female horse. So they have a lot of circulating testosterone. So that testosterone is, most of it at least in you guys and you mammals or females, are converted into estrogen. It's another hormone that it stimulates progesterone from after pregnancy. Well, not after pregnancy. Sorry, after ovulation. Sorry. Lactotropes produce prolactin. Prolactin is going to control milk production and breast development, so mammary gland proliferation in females, and it's not a tropin. And then corticotropes release 2 tropins. One's called ACTH, adrenocorticotropic hormone. All right, you notice tropin at the end, so it controls the adrenal cortex and stimulates the release of glucocorticoids like corticosterone and cortisol, which are stress hormones. There's another one released by most mammals called melanocyte stimulating hormone, or MSH, and it doesn't do much in us, normally, in humans, I should say. In other mammals, it stimulates pigment production in the melanocytes. Now, if you produce a lot of this, you have a tumor in those cells, you can produce really dark skin. But, melanocyte-stimulating hormone has a very minor role in what it does to our melanocytes. They're controlled by other factors. So those are the major tropes and what they secrete. Another image. This is actually from your physiology text, which I actually like a lot. So kind of gives you all of, there you go, insulin-like growth factors. So thrown in there for all of the different anti-pituitary hormones. I went over the posterior pituitary hormones, right? Oxytocin and vasopressin. Grouped in many different ways. Probably the best way is what they are, right? Like what macromolecule are they? And so hormones come in a variety of shapes, sizes, and forms. We've talked a lot about peptide and protein hormones like insulin, glucagon, and the pituitary hormones, right? Those are all polypeptides. They're all small proteins, typically, that are a bunch of amino acids. Steroids are also a major class of hormone, and they are all derivatives of cholesterol, as I'll show you a picture of. And then the amine hormones, or what we call biogenic amines, are amino acids that have been activated to become hormones. So when I say that, activated, that means they've been changed a little bit. Their R group has been modified, and now they are hormones. And here are some good examples of amine hormones. That's what I showed you a picture of here. Notice they're almost always derivatives of a cyclic amino acid because it has really specific three-dimensional shape, right? Which will bind really nicely to a receptor, correct? All of these that you see here are tyrosine derivatives. So you notice most of the amine hormones are a derivative of tyrosine. Dopamine and epinephrine and norepinephrine, and when you look at them, they look almost exactly the same, right? Look at dopamine and epinephrine. The only real difference between them is that hydroxyl group. Isn't that incredible? And they do really different things. Norepinephrine and epinephrine are really similar. We just remove the amino group and add a methyl group there. The two thyroid hormones, T3 and T4, are two tyrosines put together. So they're, a dityrosine. And they are named based on the number of atoms that are added to them. So notice T4 has 4 iodines attached to it. So it's called tetraiodothyronine, where T3 is called triiodothyronine because it has 3. We'll talk about why the two differences. They both kind of combine to the thyroid receptor, but we'll talk about, one has a better attachment than the other. T4 is often called thyroxine, right? Which is the major thyroid hormone. Now, all of these, I shouldn't say all of these. Number 1 and number 3 can be produced and stored either intracellularly or extracellularly pretty easily because they're proteins, right? Which a lot of the proteins are either really big or really big and hydrophilic, right? So they can't cross membranes very easily. So they're easy to store. You can store them. Steroids are not easy to store. As a matter of fact, you can't store them because they move through membranes really easily, don't they? So they have to be made real time, right? They make them real time. The cells or the glands that are responsible for producing them. I give you this image to show you insulin synthesis, but any receptor or any protein-based molecule that's going to be released from the cell is made this way, right? This shows you insulin synthesis. The big thing here, most hormones are produced as prohormones. That is, they're either a prohormone in the Golgi or stored as a prohormone in the vesicle, and when they're either changed before they're released or changed right after they're released so that they become activated. If you see pre or prohormone, that means they're in their inactive form. They can't take on their final three-dimensional shape. The big difference between a pre-prohormone and a prohormone is the pre-prohormone is the entire polypeptide sequence right after translation, completion of translation. The big problem with the pre-prohormone are there are a few amino acids that are attached to the polypeptide that are going to be removed. They were the signal, what we call the signal peptide, that told the ribosome to go to the rough ER. The docking proteins recognize it. Right? So the docking proteins that bring the ribosome to the rough endoplasmic reticulum recognize the first few amino acids that come off during translation as the signal peptide. And as the protein is dumped into the lumen of the ER, there is a peptidase here that removes the signal protein. So the whole polypeptide plus the signal peptide is called the pre-prohormone. When you remove the signal peptide or cleave it off, then the protein you end up with is the prohormone. Right? Like, for example, insulin, it's called proinsulin. Right? Proinsulin is actually a really huge protein. As you can see right here, this is proinsulin right here. And you will package it in the vesicle with an enzyme. And the enzyme, what it does is it will cleave this really long protein into actually insulin and then what's called C-peptide. And when this is released, the insulin is the active hormone. The C-peptide is reabsorbed and removed. So that's often, prohormones are going to be modified into the actual hormone. Notice insulin has a very specific three-dimensional flat shape. It recognizes its dimer very nicely. It's got a bunch of disulfide bonds that hold it together. And so the active form of insulin will bind to, is a systemic hormone, right? It binds to the insulin receptor like we talked about. Go back one sec here. In the vesicles, the major difference between the hormone synthesis and the receptor, the target cell receptor synthesis, is the protein is inserted into the membrane of the vesicle, right? So that when the vesicle adds to the cell membrane, the receptor is added to it. It's not released, right, at the surface, so that it can bind insulin or whatever hormone we're talking about here. Okay, steroids are produced real time. They're produced from cholesterol. Steroid-producing cells typically have big globules or droplets of cholesterol cytoplasmically. So, for example, when you section through a Leydig cell in the testis, you often will see a big, huge inclusion that stains brown or black under an electron microscope, and that is a droplet of cholesterol. And they use that cholesterol to make testosterone real time, because once testosterone is produced, it just simply passes through the membrane and enters the blood, right? And then it will be carried to its target. Most steroid hormones, the target receptor is in the cytoplasm, right? Because the steroid can pass directly through the cell membrane, find its receptor, and then it will travel to the nucleus and turn on gene expression directly. Again, these are just pictures of some of and again, you don't have to draw them. I just want to show you that the steroid hormones, the big difference between them is what groups are sticking off of them, right? And then where the double bonds are, which change the, rotate the shape a little bit, change the three-dimensional shape slightly. Again, look at these two. I mean, it blows me away. Now that I have kids, girls and boys, man, they're really similar and they cause really different effects in brain development, secondary sex characteristics. And they're very similar. I mean, there's the major difference is you replace this double bonded O with the hydroxyl group, and you move some double bonds around. And all of a sudden, you have two very different hormones. Right? Smart hormone, dumb hormone. Right? I can say that I'm a male in the group, right? You would agree you're often males, right? Progesterone also looks very similar to those, right? The other female sex hormones. So, interestingly, the steroids are made in a number of different ways through a huge biosynthetic pathway where you can create all the different hormones. The big one here that you need for the sex hormones are females have the expression of an enzyme called aromatase. So aromatase is what changes testosterone or any of its metabolites into the estrogens. And there are a number of different estrogens. And so without aromatase, you can't produce estrogen. Males produce estrogen. We do, actually. The estrogen is actually really important in males in the concentration of sperm in the epididymis. So we have estrogen receptors there. Songbirds, the male produces a ton of estrogen in the brain. Makes sense. It's not testosterone that makes beautiful song. It's estrogen. Right? That does it. The smart hormone teaches the males their beautiful song. So there are other places in the animal world where males do express a lot of estradiol for certain behaviors. But you can produce a lot of these all arisen from cholesterol, right? So as your major source. The other thing you probably should know is that you can produce all the cholesterol you need. You don't have to ingest any. We take in a lot of cholesterol in some of our foods, particularly poorly processed foods, right? But your body can make cholesterol. You don't need any cholesterol in your diet.